Hey everyone, it's Ms. Lopez. In this video, I'm going to go over the Chapter 6.5 notes that involve photosynthesis. So in the first part of this chapter, we talked about cellular respiration and the breakdown of glucose. In this part of the chapter, we're going to talk about photosynthesis and how that glucose is created through anabolic pathways. Photosynthesis, which is anabolic, involves two pathways. The light reactions, which convert light energy into chemical energy. In this case, we're looking at ATP and NADPH, or the chemical energy. And then the second pathway in photosynthesis are your carbon fixation reactions. This is where we're going to use the energy, ATP and NADPH, produced in the light reactions to produce those carbohydrates. So this is an overview of the process and how the two are linked. You'll notice that here are your light reactions, which do depend on light energy. So light is considered a photon, and then that light energy is going to strike the thylakoid membrane where you have an electron transport chain embedded. We're going to be able to take water, harvest electrons from that, and then use the electron transport chain to, number one, phosphorylate ADP into ATP, and then number two, we're going to reduce NADP plus down to NADPH. In this process, we will produce oxygen, they will produce oxygen as a byproduct, which then is small and nonpolar, so it can exit the membrane and then exit the cell. But the ATP and NADPH are then used to power your Calvin cycle, where where plants are going to take carbon dioxide and then use that to create sugar molecules. Light is a form of electromagnetic radiation. It is propagated as a wave, but also behaves as a particle, and a light particle is called a photon. The amount of energy in the radiation is inversely proportional to the wavelength. And what that means is, the larger the wavelength, the lower the amount of energy, and vice versa. So on this graph here, you can see several sections of the electromagnetic spectrum. The part that we usually can talk about is the section that involves visible light. You'll notice here that red has a very large 700 nanometer sized wavelength, whereas violet has 400 nanometer sized wavelength. What that means is that there's lower energy in red than there is in violet. And then anything beyond 400 nanometers is no longer detectable by human eyes and is known as ultraviolet, which has more energy and can damage DNA. X-rays have even more energy and again, damage and breaking the DNA. Whereas on the other end, on the red spectrum, anything larger than 700 nanometers is still not detectable by humans, but it's present, such as infrared, which has even lower amount of energy, and then microwaves and radio waves, which have lower amounts of energy still. Photons can be absorbed by specific receptor molecules, which are raised to an excited state. And that excited state is considered to have more energy. So the molecule that's absorbing this photon has energy in it. And when that molecule absorbs that photon, it goes from what is known as the ground state and absorbs that energy and becomes excited. So the excited state of a molecule has more energy than the ground state of a molecule. Pigments are molecules that absorb wavelengths in the visible spectrum. Now, the most common pigment that students know is chlorophyll. Chlorophyll is going to absorb blue and red light, and then the remaining light that it doesn't absorb is green. So by reflecting or not absorbing that green light, this is why we see plants as the color green, because chlorophyll is absorbing the red and blue, but reflecting back to our eyes the color green. The absorption spectrum is the plot of energy absorbed against the wavelength. But the action spectrum is the plot of biological activity of an organism against a wavelength. So we can use these spectrums, whether it's the absorption or the action spectrum, to see how active, how much light a pigment or molecule is absorbing versus at which wavelengths is that molecule or particle acting, becoming excited. In plants, there are two types of chlorophyll, chlorophyll A and chlorophyll B. Now, chlorophyll A is your main type of chlorophyll, and chlorophyll B is what's known as an accessory. It helps out to increase the range of light that chlorophyll A is going to absorb. Now, these two together will absorb red and blues, and then what's going to happen is that these two combine will reflect both the little bit in the yellow and green range, which is why we see plants as green. But there are other accessory pigments as well, and what they do is they help support by absorbing even more light than these two chlorophylls would be able to absorb on their own. So we typically see chlorophyll A and B, as a green pigment. That means it's reflecting the green light. But hidden, there's so much chlorophyll in this leaf, and hidden behind the green are two other pigments. One of them is called xanthophyll, which is kind of yellow in its color, so it's reflecting yellow light. And then your carotenoids are more orange color. They're reflecting the orange light back at us. So there's so much in the spring and the summer, there's so much chlorophyll in green plants that you can't see the xanthophyll and carotenoids. But in the fall, when plants begin to shut down, they stop producing this chlorophyll, and then what starts to show when the chlorophyll starts getting broken down, what you start to see when the leaves change color in the fall, is those xanthophylls and carotenoids that remained and that were always there. We just couldn't see them because of how much green pigment there was. Anthocyanin is more of a red color, and this is not there all year. Anthocyanin is not produced until the chlorophyll begins to break down. The pigments are arranged into light harvesting complex or antenna systems. A photosystem spans the thylakoid membrane in the chloroplast and consists of multiple antenna systems surrounding in a reaction center. So let's take a look at what this looks like. So you have here your photosystem, which is going to act like, think of it like a satellite dish that's collecting light and redirecting it towards this reaction center. So each of these chlorophyll molecules are going to be able to pass on that energy until you get to your main reaction center, where there will be an electron that's going to become excited. So this electron is going to go from a ground state to an excited state, and this is all embedded in that thylakoid membrane of the chloroplast. So this excited state, this excited electron is then donated to an electron acceptor so that the electron can get passed along an electron transport chain. The electron acceptor is the first carrier in the thylakoid membrane, or in the electron transport system in the thylakoid membrane. Your final electron acceptor at the end of the electron transport chain is NADP+, which is going to get reduced down to NADPH. In this electron transport chain, we're also going to produce ATP because we can create a chemoosmotic gradient. We can create a concentration gradient on either side of the thylakoid membrane, similar to the way that we did when we talked about oxidative phosphorylation. Although in this case, this ATP is going to be referred to as produced by photophosphorylation. So here is the light-dependent reactions that involve that electron transport chain that we covered in our notes. You have your first reaction center, which is called your P680, which is going to get electrons from water. So we're going to the cells are going to split water to harvest electrons, and then this oxygen is produced as a byproduct. That electron is then going to be excited by all the energy that's captured and redirected and focused onto this area, and then that electron is going to be transferred to the next transmembrane protein. Now this is an actual transmembrane protein similar to what we saw in oxidative phosphorylation. Once it receives that electron, it's going to pump hydrogen across the membrane. The job of the photosystem, in this case photosystem 2, is just to harvest that electron and to focus the light on that electron so that it can become excited. Once the electron has been donated to this protein, this B6F, the protein is going to change its shape, pump the hydrogen, and then the electron will get passed on to the second photosystem. Thanks to the hydrogen that was pumped across at B6F, we're creating a hydrogen ion concentration gradient, and then ATP synthase, the exact same enzyme we saw in oxidative phosphorylation, can then produce ATP by phosphorylating ADP as the protons are moving down their concentration gradient. This electron is going to get excited at this reaction center called P700, and then that excited electron is going to be carried and donated to an enzyme called NADP plus reductase, which will take that NADP plus and reduce it down to NADPH. The ATP and NADPH produced in the light-dependent reactions can now go to the stroma and help produce the carbohydrates in the dark reactions, or what is known as your Calvin cycle. So ATP is needed for those carbon fixation pathways. The non-cyclic-like reactions would not provide enough ATP. So sometimes what happens is a phenomenon known as cyclic electron transport. Cyclic electron transport only uses photosystem 1 and produces only ATP. As an electron is passed from an excited chlorophyll through the electron transport chain, it is recycled back to the same chlorophyll molecule and then re-excited. So this is called cyclic photophosphorylation because it's the same electron going kind of in a cycle. So you have an electron here at P700, which becomes excited. The excited electron then is transferred to an electron carrier, which takes it back to the B6F. B6F is going to pump a hydrogen across the membrane, and then the electron is going to come back to photosystem 1, where it's going to get re-excited. That re-excited electron gets transferred back to the B6F, which is going to pump another hydrogen across the membrane. And so the electron goes somewhat in a cycle between your photosystem 1 and the B6F complex. Every time the B6F complex receives the electron, it's going to pump a hydrogen across the membrane. That's going to increase your hydrogen ion concentration gradient on one side of the membrane, and because of that high concentration gradient, ATP synthase is now going to be able to produce ATP by phosphorylating ADP and inorganic phosphate. As this high proton gradient, as those protons are moving back through ATP synthase, we're going to generate even more ATP. Now, once you have enough ATP, you can move forward with the Calvin cycle. In the Calvin cycle, the energy in ATP and NADPH is used to fix carbon dioxide, which is a gas, into a reduced form to eventually form carbohydrates. This is going to occur in the stroma of the chloroplast, and each reaction is catalyzed by a specific enzyme. This biochemical pathway is a cycle and is composed of 3 distinct processes. In the first phase is taking that carbon dioxide and attaching it to an organic molecule of RUBP. Once that carbon is attached to that RUBP, this is known as your carbon fixation phase. Once it has been attached, it's going to break. And what we're going to do is we're going to start the next phase right here, which is your reduction and sugar production phase. In this case, what's happening is we're going to have to input some energy in the form of ATP, and we're going to need electrons that were generated from the NADPH. And we're going to reduce this molecule until we get the sugars produced. Now, this doesn't produce glucose right away. We do have to regenerate our starting molecule, which is the last phase. We do have to regenerate that starting molecule. And so we're going to slowly add to this building sugar 1 piece at a time. So it's going to require 6 turns of the Calvin cycle to produce 1 carbohydrate. Thanks so much for watching, guys. Bye.